Swayam Prabha. Digital India. Educated India.
So, we continue with the discussion of the DNA and the RNA structures, and you see here, as I said, the ribose ring or the 5 membered ring is the important element. What are the elements of the DNA structure? Well, the phosphate backbone is one, and the second thing is the sugar ring, and the sugar ring, as I said, is a 5 membered ring here. Now, we have to define the structure of the sugar ring. So, and that is an important element. We have to define the structure of the phosphate backbone, the various angles which are present here, the torsion angles which are there, we will come to that. And then you have this sugar ring. The sugar ring structure is determined by these torsion angles which are indicated here. So, nu0, nu1, nu2, nu3, nu4, not all of them are very independent. The torsion angle is defined in this manner. So, the nu0 torsion angle is defined as the angle around this bond here, rotation angle around this bond, which defines the relative positions of C4 prime and C2 prime. Okay. C4 prime, O4 prime, C1 prime, C2 prime, and the central around the central bond there is this torsion rotation angle. So, these are also called as dihedral angles, because these define the angle between the two planes formed by these groups C4 prime, O4 prime, C1 prime. These atoms form one plane, and O4 prime, C1 prime, C2 prime, these form another plane, and it is the angle between these two planes that defines the dihedral angle, or it is also the torsion angle around this. So, likewise you have this in U1, which is O4 prime, C1 prime, C2 prime, C3 prime, and then you have Nu2, which is C1 prime, C2 prime, C3 prime, C4 prime, and Nu3, C2 prime, C3 prime, C4 prime, O4 prime, and you have Nu4, C3 prime, C4 prime, O4 prime, C1 prime. But however, notice that all these torsional angles are not independent, they are dependent on each other, because the ring is closed, and then there are also important, you can see do physical chemistry calculations, one can see how many degrees of freedom are there. So, you have the in a typical one you have the 4 bonds and 3 bond angles, and you have 2 torsion angles. Typically 2 torsion angles are sufficient to describe this, but then once you have the ring closure, then you actually have only 1 torsion degrees of freedom left, and that is what is indicated in this picture here. You see, these ones are all related, all these torsion angles are related in a particular manner, and the 1 degree of freedom which is there, which is called as the P. When one defines this P, this is called as the pseudo rotation angle. The pseudo rotation angle, depending upon the value of the pseudo rotation angle, so you have different kinds of nu i's here. And this nu I is the individual torsion angles, is given by tau m. This defines the maximum distortion from the plane of the ring, how much a particular atom is going out. So, this is the kind of a quantity number which indicates how much is a particular atom going out of the plane, what is the maximum value. But this will have a certain constraint, because if the bond angles and the bond distances are well fixed and they cannot be changed too much, and therefore this actually is a kind of a well defined element. This is typically of the order of 38 degree. Okay, and then the cosine, the P is a value which can take values from 0 to 360, and I minus 2 delta, delta is a fixed number like 144, you put delta is equal to 2 and keep varying I through different values, then you will see by you get different kinds of sugar geometries. You depending upon the value of P, you get different geometries, and all of these are indicated in the kind of a circle, because the P can go from 0 to 360, or you can say it goes from 0 to minus 180 or plus 180 and so on and so forth. Okay. And these are typically indicated what is the kind of a structure that we will get. These are all got certain labels, that is what is the kind of a structure the sugar ring has got. And here you see, if you make a plane of a particular thing, put the 3 atoms in a plane C1 prime, O4 prime, C4 prime, if you put them in a particular plane, what is the disposition of the 2 prime and the 3 prime atoms with respect to that plane, and then you will get different, and the C4 prime is always on one side. So you get different kinds of structures for the sugar ring. So in this case the 2 prime is above the plane of the ring and the 3 prime is below the plane of the ring, and the O4 prime is here. The O4 prime is always there only. And in this structure the 3 prime is above the plane of the ring and the 2 prime is below the plane of the ring. So here you see both the things are above and below. So you have the extent of the things which are above and below. So you have the 3 prime above, 2 prime below, but the degree is more. So 3 prime less, 2 prime more, the degree is less, and therefore you get different kinds of structures which are indicated by typical CN. This group of structures which are indicated where the 2 prime is above the plane of the ring, these are called as S conformations or called the south, and this is the south here. This is the south in this circle, this below this central line, this is the south and this is the north. So all those conformations which are below here, these are called as the south conformations, the S conformation, and these are characterized by the 2 prime atom going above the plane of the ring, and these are called as C2 prime endo. If it is above, it is called as endo, anything which is below, it is called as exo. And therefore you have here in these structures, then you have, see, when it is this calls in the south, you have the C2 prime endo. The C2 prime endo is this one here, okay. So all of these are C2 prime endo, and to different degree how much is the 3 prime below. So you get what we will say here C2 prime endo, C3 prime exo, but the degree of the 3 prime which is below or the degree to which this one is above, that determines the different positions along the in this circle here. Therefore you see here this is 0 to 180. On this side similarly what we have the north conformations, it is the 3 prime which is above the central line, okay, and this is the 3 prime endo, okay. Therefore this is the 3 prime endo here, and if you take the p value, how the p value is changing, so this goes from 0, 36, 72, okay, this becomes 90. So at this 90 the O4 prime endo, now the other things becomes that the O4 prime is actually going out of the plane, and that is not shown there, okay. So your O4 prime becomes above the plane, and put the other 3 atoms in the plane of the ring, in a particular plane, and then you have the twisted conformations. They are described as endo conformations, twisted conformations. Twisted means both the things are above and below, but to different extents, it is not only one. So you have in one particular case then only one, which is all the 4 atoms are in a plane and only one atom is above the plane. You can also think about that, and those ones are these O4 prime endos, O4 prime endo or O4 prime exo. 4 atoms in a plane and one of them is above, and all other ones are kind of twisted to a certain extent, okay, which is closer to this to 180. This is called as C2 prime endo, and then you have C3 prime exo here and so on and so forth. So you see P is an important parameter which determines the nature of the sugar ring, and the sugar ring varies from one nucleotide unit to the second nucleotide unit. It can vary. It can vary in the DNA and the RNA. Why it can vary? Because the C2 prime here in the case of DNA has 2 hydrogens here, whereas in the RNA it has 1 hydrogen and 1 oxygen, and therefore this can create a kind of a steric differences and interaction differences between the oxygens here and between the one nucleotide unit to the next nucleotide unit. Therefore this sugar ring can have different conformations. Indeed you will find, as we will show later, that in the case of DNA this sugar ring adopts the south conformations by and large. The C2 endo conformations is called as C2 prime endo conformations, and in the RNA they adopt what is called as the C3 prime endo conformations, and that primarily comes because of the oxygen at this point. Okay. So now you see what it reflects in the case of there are different kinds of structures, and these are classified as A, B, C, D, E in the DNA canal. Now the fiber diffraction data could not give you any more detail about this. Now how do we get this information? So, you can use tools like NMR or crystallography, and people then actually synthesize small DNA segments to get more details into the structures of the individual nucleotide units or short segments of the DNA segments, because there is a repetitive in it. So, therefore, you can say if you take a short segment of the DNA segment or the RNA segment, you might be able to think what is the kind of a structure which is propagating through the entire polymer. And that is what was done, and they came out with the different kinds of DNA structures, and those ones are indicated here. And this is the stereo 12 base pair of A DNA. This is a particular kind of a DNA structure where it is an A DNA. Okay. And this is the structure of this molecule, deoxy GGTATACC. Okay. And this is the 5' end here, and this is the 3' end here. The sequence goes as GGTATACC. This is how the DNA sequences are written. Okay. And here you see the structure has certain kind of a fat structure, and the base pairs are tilted with respect to the axis of the DNA, axis of the double helix. The axis of the double helix may go like this, and the base pairs are tilted. In this half they are tilted like this, in the top half they are tilted like this, and that typically the fiber diffraction data had shown that one complete turn of the DNA. So, what is the complete turn? So, you go from here to here like this, and then you come to the beginning, and that is one complete turn. From here to one complete turn, and the fiber diffraction data had said that there can be approximately 10 base pairs in a particular segment. Okay. And depending upon the structures, of course, there can be slightly more, and also in the later segments which have been discovered. Therefore, you have this a particular half and this particular half, and this structure is called as the A DNA, because this DNA molecule has structures which are very reminiscent of the 3' endo sugar geometry, and therefore this is called as the A DNA. And normally what is present in the larger probability in your solution inside the cell is what is called as the B DNA, and this is the B DNA. And this is the structure obtained by a larger DNA sequence under the different conditions, and depends upon the hydration level also inside your structure, inside your DNA a crystal, how much water molecules are there. So, it also depends upon those ones, and this is the structure of a 12-mer DNA segments which has the base sequence CCAA CG TG TT GG. Okay. Notice here this is self complementary. What is the meaning of self complementary? So, if I write here the sequence CC AA CG TT GG. Now, this is the 5' end and this is the 3' end, right. Now, what I do, I write the same sequence here back, CC AA CG, then I have TT GG. So, this is the 3' and this is the 5'. You see, I have written the same sequence twice. So, therefore, this is called as the self complementary sequence, and therefore, one molecule which is there, naturally it goes into the form of a double helical structure like this, and this is easier to work with such molecules. Otherwise, you will have to generate 2 different kinds of sequences and then to make a duplex structure. So, this molecule was chosen, though it automatically forms the duplex structure as the most stable form. Okay. So, this is self complementary. Okay. And this molecule, the structure showed was like this, and it is little bit more elongated, you can see, compared to the RNA molecule and the A DNA. This structure is called as the B DNA. Okay. And it has clearly 2 different kinds of grooves. One can identify 2 grooves. There is one groove which goes like this, which is called as the major groove, and there is another groove which is the smaller groove, which is called as the minor groove. So, there are 2 different kinds of grooves described for the DNA structure. So, this amount, depending upon the amount of space which is available, and you have a groove which is defined. Okay. The base pairs open in the 2 different grooves. One side of the base pair opens in the major groove, other side of the base pair opens in the minor groove. Okay. So, that is when you explicitly write the base pairs, and that is what one sees. Now, this is the third type of DNA segment, and naturally, okay, now let us go behind here. Now you see these ones are right handed helices here. You see this is one right handed helix. The helix goes like this, okay. This is the right handed helix, and the other one is coming in the opposite direction. So, that is also a right handed helix. 2 right handed helices are coming like this, and then they are held together by these hydrogen bonds in the base pairs. Okay. So, in this BADNA as well as in ADNA, we have 2 right handed helices which are intertwined to form base pairs, and that is, but there are differences in the structural elements. How much is the distance between one base pair to another base pair? Typically it is about 3.64 angstroms here. So, therefore, if I take 10 base pairs, you will have the one end of one's complete this turn of the DNA is about 34 angstrom. This is what I used earlier for the calculation as to what is the total length of the DNA segment. Okay. And therefore, you have this, the unit rise is 3.4 angstroms, and then you have a 34 angstroms in the DNA. In the case of RNA is slightly smaller, and the number of units also is given the same, the height is slightly smaller. Okay. Now this is another kind of a DNA structure which was discovered. This actually came quite late. This came somewhere in the close to the 1980s, 1979, and this is called as the zDNA or the zDNA, and this actually is a left handed helix. Okay. This is not a right handed helix, this is the left handed helix, and this very characteristic feature of this is that it is not a monomer which is repeating here, but it is a dimer. So, the dimer structure is the basic unit of this repeating unit, and this is the left handed DNA structure, and it occurs only in such point of sequences which are rich in CG segments. CG segments, initially it was not clear whether it is actually biologically present or not. This is called as the zigzag nature of the phosphorus phosphate residues that you can see. So, this is also, that is why it is called as zDNA. It is zigzag nature. It is not a proper helix as you can see in the case of BDNA or the ADNA, but this is a zigzag nature, and the dinucleotide is the repeating unit, unlike the mononucleotide which is the repeating unit in the A and the BDNA, and in the dinucleotide the 2 units are different structural features, and that is an important factor how they become different. And these are typically observed in very high concentration of whenever there is high concentration of salt. And why did they do it? Of course, just curiosity, and they discovered this, and then of course it became an important point. Okay. So, here is the summary of all of these DNA segments, the structures of the different DNA segment. So, here you have the ADNA, then you have the BDNA, and this is the crystal structure which are these are from the model. Okay, ADNA, the small a which is here, this indicates from whatever the model they built, the Watson and Crick, these are from fiber diffraction data, and these are from single crystal structure. GGCC, this is also, you notice, is a self complementary sequence, and this forms an ADNA. The BDNA, this form which is there, CG, AATT, CG, this is also a self complementary sequence, because if you write in the similar reverse way, you will find it is the duplex. And the zDNA is, as I said, CCGGC, that was the structure which was there, and it is a dinucleotide which is repeating unit here. You have the C residues and the G residues, they have different structures. The individual units are different structures. Now what are the structures here? What are these alpha, beta, gamma, delta, epsilon? These ones, these are the torsion angles along the backbone of the DNA. So you have this backbone phosphodiester linkages all over there, and that is the various 6 torsion angles which are present along the phosphodiester backbone, and this chi is the angle which connects the sugar ring to the base. Sugar ring to the base, that is the C1 prime nitrogen, that bond, the torsion angle around that bond, that actually defines what is the orientation of the base with respect to the sugar ring, and that angle is called as chi torsion angle. and you notice here these have certain ranges of values, typical ranges of values by and large. So these ones, the alpha is around between -50 to -75, and BDNA it has -41, this is slightly lower compared to these ones, and the beta angle, this is 172 to 175, and here in the BDNA it is smaller, 136. In the gamma delta, these are along the backbone. So along the polypeptide backbone you have these different torsion angles there. So if I want to draw here, this is the CH2, this is the 5 prime, this goes to the oxygen here, this is the 5 prime, then I have the C4 prime, the C3 prime, then the O3 prime, okay, then you have the CH2 again, this is the 5 of the other end, okay, and then you have the P here, then you have the O here again, and your sugar ring is here. This goes to the O here, C1 prime, C2 prime, and this is connected to this, and this angle is the delta torsion angle there, okay, and this is the gamma, and this is the beta, and this is the alpha, and this angle is the epsilon, okay, and this is your xi, and the one which connects to the C1 prime to this on there, that is the chi, that one angle which is not, there is no space here, and this goes, this is the chi here, okay, this is the 5 prime again, CH2. So therefore you have this alpha, beta, gamma, delta, epsilon and xi. So these are the 6 torsion angles what you have in this here, okay, and how these values are changing. And of course there is also people did a DNA, RNA decamer, and of course this is a mixture of the DNA and RNA, and this has different kinds of torsion angles here, the values are indicated there. And the aRNA is the sugar ring is more C3 prime endo here, there is a sharp contrast, and what it reflects, which angle reflects the sugar ring here, this by and large it is in the delta, because the delta is in the sugar ring, C4 prime, C3 prime, that is part of the sugar ring, and so it is reflected in the delta torsion angle how these ones are different, okay. You see here the aDNA has 7991 and the bDNA has 130 between 120 to 140. So therefore this is quite characteristic, and the zDNA has alternating this and this. So you have the C residues has this C2 prime endo, and the G residues has the so called C3 prime endo. So if the delta is around the 7990, that is the C3 prime endo structure, and if it is around 120 to 140, that is the C2 prime endo structure. So therefore in the zDNA you see the C residues and the G residues have different sugar geometries. So one of them has the C2 prime endo kind of a thing, other one has a C3 prime endo kind of a structure, okay. And the beta torsion angle is mostly around the trans value, and except for this particular one, here it is 136, otherwise it is mostly around the 180 value roughly. And similarly this is the gamma is roughly around the 40 between so called the Gauss conformation, except for here the G residues in the zDNA, and this goes into the trans area. So this is -169 is like the trans area, all of these ones are so called Gauss conformations here, close to less or more and things like that, okay. So this is how the various structures, now all this information has come from the single crystal structures of short DNA segments, okay. Now there is also one should describe it, what about the base pairs? Base pairs, are they all parallel to each other or they are different from one another? The base pairs can differ to, and you have to describe what is the relative orientations of the bases in the base pairs. So you can define that in with different parameters here. So these are the various parameters which are used to describe the base pairs. I told you that the base pair which is there on one side of that is the minor groove, other side is the major groove. So typically the base pairs open on the two sides, because these are in the interior of the double helix and they open on the two sides of the double helix. So one side is the major groove, other side is the minor groove, okay. So and now you see here it is the 5 prime to 3 prime, it is going here, this is 5 prime to 3 prime, and the base pairs are held together like this, okay. And now are these in the same plane or they can be tilted. Are they parallel to the orientation with respect to the double helical axis, or are they tilted with respect to each other? Various kinds of parameters can be described here. What is the orientation with respect to the axis of this? Here it is perpendicular to the helical axis. Here it is slightly tilted in one dimension, one direction, okay. This tilted with respect to what? So, this particular axis along this. Now, here it is tilted with respect to the other axis, okay. So, there is a rotation here, this is indicated by the rotation here. You indicate a rotation, consider this as a planar one, and in the whole base pair is the plane, and you take a rotation around this axis, therefore this becomes like this. Now you take a rotation around this axis, it becomes like this, okay. So, therefore you can describe the orientation of this base pair using this one particular parameter, that is when these are all parallel, if they are parallel. Now, but whether they are all exactly in the same pair like this, or there is a small opening here, okay. So, you can again describe these ones here, there is an opening with respect to this axis, their two bases are like this, or the two bases are like this, or the two bases are like this. So, this is the different torsion, this is the parameter which is called as the kappa here, and you have different, these are labeled by different parameters, and you can describe this as within the same base pair how they are oriented with respect to each other, the two bases are. Now, you can also describe the relative orientation of two base pairs, consecutive base pairs. Two consecutive base pairs, if you look at these two consecutive base pairs with a rotation around this particular bond at this particular axis will displace the two base pairs in this manner, okay. So, therefore not exactly sitting on top of each other, but slightly displaced with respect to each other, their planes, okay. Now, if they are angled like this, then difference is called as the roll, this is called as the twist, this angle is called as the twist, this angle is called as the roll, and this angle is called as the tilt. So, you differentiate these different parameters by different names. So, one of them you call as the twist, this is called as the roll, this is called as the tilt, and similarly here you call them as opening, and this is the propeller, and this is the buckle. So, here also inclination, these ones are called as tip, this is inclination, and this is the coordinate frame in the same, indicating the coordinate frame, and all of those ones are described here in a little bit more explicit manner. So, you have the summarize. So, you have the minor groove on this side and the major groove on this side. So, if the base pair is like this, you draw an axis like this through the center of the base pair, and then you have a vertical one and you have an axis like this. You define x, y, z, these three axes with respect to the base pair, and you can define the positions of all of these groups as rotations with respect to these three axes there, and that is called as the slide, and then you have the shift. So, how they are shifted with respect to the center, okay. So, whether it is exactly going through the center or it is shifted with respect to that. So, this one is shifted. So, this is a kind of a slide and this is a shift. So, these are the different parameters which are used to describe the orientations of the base pairs in the same in the plane, and so you have so many parameters to describe the structure of the DNA segment. So, you have the sugar geometry on one hand, then you have the torsion angles along the polypeptide backbone, and then you have the base pair orientations with respect to each other, and whether they are in the same plane or they are slightly tilted with respect to each other, and how are the stacking, whether the base pairs are stacking exactly on top of each other or they are slightly different from one another. So, these are the various parameters. All this information has come because one could obtain high resolution structures of small DNA segments, and this is taken from this paper of R. Dickerson. This published in 1989. So, this is the description of the individual base pairs here, average helix parameters for the major DNA conformations. Okay, we need not go into the details of these numbers here, but this is a kind of an indicator as to what sort of values are there for the different parameters characterizing the DNA structure in the different DNAs. So, depending upon the small variations which were present, so different kinds of labels have been given for the different DNAs. We talked about the A DNA, B DNA, but there are also some variations which occurred and they led to what are called as the C DNA, D DNA, T DNA. Then you also have the Z DNA which you also described in major detail, and then you have the A RNA and A prime RNA. These ones are the RNA segments, all others are the DNA segments. These are only minor variations with respect to the major ones. The ones which are by and large available in the natural sequences are the B DNA and the A DNA, and even there the RNAs are mostly in the A form and the DNA is in the B form, and this depends on also on the extent of hydration, how much is the water content, depending upon that you get slightly different structures. Okay. And this is the continuation of the same. So, what sort of parameters are there for the base pairs, orientations of the base pairs in the, and this is for the Z DNA, and those ones were for the A, B, D and C and D etc. And since in the Z DNA you have different things for the two different steps, the CG steps are different, therefore you have different parameters here for the two things. So, in other words, we need to characterize this kind of details for the structure when we want to calculate the structures of the molecules, and crystallography has been used and NMR also has been used, and we will see more what kind of structures have been determined by the NMR data, and these happen in the solution. Okay. So, this has become possible with various kinds of NMR experiments and certain algorithms which have been developed. So, I think we can stop here.